Hereby, I open this academic ceremony in which Alžbeta Chabroňová will defend the academic thesis Chondrocytes, Ribosomes in the Spotlight. May I invite you to present a summary of your study and the conclusions of your thesis.
Well, thank you, highly esteemed prorector. Dear members of Corona, my supervisors, colleagues, friends, and family, thank you very much for being here today, either in person or online. And in the following 15 minutes, I will be giving a short summary summarizing my doctoral research on the topic of ribosomes in chondrocyte health and osteoarthritis. So, movement is an essential part of life, and it is like breathing. We don't think about it too much, we just do it, until there is a problem. Worldwide, more than 500 million people experience unbearable pain doing simple movements or just walking few steps. These people suffer from osteoarthritis. Osteoarthritis is age-related degenerative joint disorder, and it is a leading cause of pain and disability in adults. It is a disease of the whole joint, and all of its tissues are affected, including articular cartilage, synovium, synovial fluid, and subchondral bone. Unfortunately, at this moment, there is no cure for osteoarthritis. We can manage the symptoms of early-stage osteoarthritis by using over-the-counter painkillers or anti-inflammatory drugs. And when osteoarthritis reaches its end stage, patients undergo joint replacement surgery in which the damaged and osteoarthritic joint is replaced by prosthesis. This is, however, very invasive and also risky surgery. And more importantly, the artificial joint has only a limited half-life. So after several years, patient needs a revision surgery, and that's even more demanding. So joint replacement and artificial joints are really not solutions to osteoarthritis. What we need is to target the biology behind the disease, and then we might be able to slow down or reverse its development. In this thesis, we focused on one of the hallmarks of osteoarthritis, which is degeneration of the articular cartilage. Articular cartilage is a proteinaceous layer that covers and protects the bone or the ends of the bones within the joint. It is composed of the proteins that create the cartilage matrix. The proteins of the cartilage matrix are made by chondrocytes, which are highly specialized cells that are located within cartilage, and they contain millions of ribosomes. Ribosomes are basically cellular machines that translate or they decode the genetic information which is encoded in mRNAs, and therefore they synthesize proteins. Once ribosomes are done with translating and synthesizing proteins, the chondrocytes secrete these proteins into their environment, and therefore they create this protein-rich extracellular matrix. During development of osteoarthritis, healthy chondrocytes change and they become osteoarthritic. These osteoarthritic chondrocytes that then produce enzymes that actively digest and degrades the healthy proteins in the cartilage matrix. And further on, these digested healthy proteins are replaced by osteoarthritic ones, which are also produced by these osteoarthritic chondrocytes. So, altogether, the cartilage protein composition is changing, and with that also its biomechanical properties. And gradually, the cartilage matrix degenerates until there is basically hardly any cartilage left to protect the bones, and the movement becomes painful. So I mentioned before that osteoarthritic chondrocytes produce different proteins than healthy chondrocytes. And this is because during development of osteoarthritis, chondrocytes change the genetic program and they just transcribe different proteins, meaning osteoarthritic proteins, therefore generating osteoarthritic mRNAs, which are then translated by ribosomes into osteoarthritic proteins. And despite the fact that ribosomes are really instrumental for this production of osteoarthritic proteins, they were never really investigated in osteoarthritis. And this is mostly because it was believed that ribosomes are just passively translating whatever mRNA they encounter, whether it is healthy or osteoarthritic. However, new research data are showing that this is really not true. And in fact, ribosomes really actively regulate translation, and therefore they can contribute to disease development. So in this thesis, we focused on ribosomes, and we investigated the roles of ribosomes in chondrocyte health and also osteoarthritis. We looked into 4 different aspects related to ribosomes. Starting with ribosome biogenesis, which is basically a process in which chondrocytes make ribosomes. And we were interested to learn whether the process of ribosome biogenesis is affected in chondrocytes during osteoarthritis. Next, we looked at a ribosome composition, because not all ribosomes are built the same. And we looked whether the composition of ribosomes of osteoarthritic chondrocytes is different than from healthy chondrocytes. We also looked at ribosome modifications, which are these special modifications that ribosomes can have. And we were again interested to look into whether osteoarthritic ribosomes are differently modified than healthy ones. Lastly, we investigated ribosome activity and function and whether it is regulated in osteoarthritis. Also, we looked whether ribosome changes in ribosome compositions or modifications affect ribosome activity. So, starting with ribosome biogenesis, which is, as I mentioned, the process in which individual ribosome components are assembled into fully functional ribosome. And this is a very complicated and also intricate process. And as such, it needs help of hundreds of ribosome biogenesis factors. An important class of ribosome biogenesis factors are small nucleolar RNAs, or SNO-RNAs. So we started with investigating the role of SNO-RNA RMRP, which is an important factor for ribosome biogenesis. For these experiments, we took dermofibroblasts from patients with mutated RMRP, as well as matched controls. And we transdifferentiated these fibroblasts into chondrocytes and investigated these. We found out that SNO-RNA RMRP is an important factor for chondrocytes health. And basically, it regulates chondrocyte status, so whether the chondrocytes will stay healthy or if it will become osteoarthritic. Going further, we utilized a bit more broad approach, and rather than focusing on a single SNO-RNA, we investigated all of them. So for this, we took cartilage biopsies of a healthy cartilage from young individuals, as well as biopsies from undamaged and damaged or osteoarthritic cartilage from the knee that is suffering from osteoarthritis from the patients who are waiting for joint replacement surgery. And then we measured levels of SNO-RNAs in these 3 types of biopsies. We found out that as chondrocytes age, they change levels of specific SNO-RNAs. And these changes in SNO-RNA levels then promote the development of osteoarthritis. So altogether, we showed that osteoarthritic chondrocytes have different SNO-RNA profiles than healthy chondrocytes. And this have consequences beyond ribosome biogenesis, because what I didn't mention is that SNO-RNAs also guide ribosomal modifications. So differential expressions of SNO RNA in osteoarthritic chondrocytes suggested that these ribosomes of osteoarthritic chondrocytes might be differentially modified. So to investigate this, we took healthy human chondrocytes and we cultured them for 14 days, either in the presence of regular control medium or in the presence of osteoarthritic synovial fluid, which was taken from the patients suffering with end-stage osteoarthritis, waiting for the joint replacement surgery. After 14 days, we isolated and analyzed the ribosomes, and we found out that indeed ribosomes of osteoarthritic chondrocytes are modified differently than ribosomes of healthy chondrocytes. And what is the most important is that these osteoarthritic ribosomes function differently. So they less efficiently translated osteoarthritis inhibiting mRNAs, while more efficiently translated osteoarthritis promoting mRNAs, and therefore generating more osteoarthritic proteins. And lastly, we were interested to find out which factors that are present in articular cartilage or synovial fluid, for example, can actually regulate ribosomes. For this, we investigated TGF-beta, which is a cytokine that is present in cartilage, where it can bind to receptors on the surface of chondrocytes and therefore trigger certain responses. And these responses could either promote chondrocyte health or they can lead to osteoarthritis. So to investigate the effect of TGF-beta on ribosomes and translation, we used human chondrocytes that were cultured for 3 days, either in the presence of the regular control medium or in the medium supplemented with TGF-beta. After 3 days, we isolated proteins that were secreted by these cells, proteins that were present within the chondrocytes, and also ribosomes of these control or TGF-beta treated chondrocytes. And we discovered that TGF-beta regulates ribosome biogenesis, also ribosome composition and ribosome modifications. And importantly, it also affects ribosome activity, where it increases the protein synthesis of chondrocytes. So for the conclusions, in this thesis, we investigated ribosomes. So far, very neglected components of chondrocyte biology. And we showed for the very first time that they are important players controlling chondrocyte health, but they also play a role in pathobiology of osteoarthritis. And we could use this knowledge to, for example, design ribosome targeting drugs, and in this way force chondrocytes to produce healthy proteins and therefore restore cartilage matrix and treat osteoarthritis. So this was a short summary of my research. With that, I thank you all for attention. I thank Prorector for giving me the opportunity to present the results and conclusions, and I'm giving floor back to you.
Thank you very much for a very clear presentation. We now start with the opposition, and the opposition will be opened by Professor Schroen, Professor of Experimental Cardiology at Maastricht University, and she was chair of the assessment committee.
Thank you. Dear candidates, thank you so much for this beautiful overview of your work. I would like to congratulate you on your thesis, which is fantastic, I thought, and also congratulate your promotion team with the beautiful work. I would like to first ask one of your panelists to read proposition 2.
Well, thank you, highly esteemed opponent, and I guess...
Human articular chondrocytes respond to the osteoarthritic microenvironment by functionally adapting their ribosomal pool.
Thank you so much. Now my question is, what kind of adaptive response is that? You say in your proposition it's a functional adaptation, and I always was taught from my background that an adaptation comes for a reason, evolutionary reason, or Did we benefit from any kind from this response? Can you speculate a little bit on that?
Yeah, definitely. Okay, so thank you, highly esteemed opponent, again. Thank you for your compliments for the thesis. And for the response, so yeah, I'm saying about adaptive mechanism, and I think this is yeah, pathological adaptation. So it is related to the disease process, which osteoarthritis is, and yeah, well, also all the results from my thesis are suggesting that these chondrocytes are slowly selecting maybe their ribosomal pools and their ribosomes to select for these osteoarthritic ones to promote the disease progression. So I think that is, yeah, adaptation towards pathology in this case. But of course, this mechanism is universally applicable, I think, for all types of processes within the cell. So I would definitely expect that this sort of adaptation will play a role in development, for example, or different developmental stages, or yeah, during chondrogenic differentiation, for example, which is important for our growth. So
okay, so that does sound a bit smarter than an adaptation to a pathology that you don't want, actually.
Yeah, definitely.
Yeah, I would like to move to another topic, which is related to figure 2a, which was for me very helpful. It's on page 11. It's a very simple figure, probably for you, but for me, as I'm not in the osteoarthritis field, and actually never really gave the composition of bones that much thought. I appreciate that most chondrocytes in this figure are not in contact with each other. Is that true?
Yeah,
especially in this layer that is relevant for osteoarthritis.
Yeah, in the healthy cartilage, the contact between chondrocytes, well, they are not in direct contact in healthy cartilage.
Mm-hmm.
I mean, they are also surrounded by this pericellular matrix and then territorial matrix, and yeah, they are very sparsely distributed in the cartilage, which maybe might be a little surprising.
Indeed, yeah. Now I was wondering, because nowadays we are thinking about animal-free experiments, and you have been using human cells. And I was wondering, how was the confluence of these cells? Are they in contact with each other, or how did you culture them?
Well, we cultured them, so the seeding density was approximately 30,000 cells per square centimeter. So they were, I think, maybe more in contact than in the natural occurring cartilage in our in vitro experiment.
Indeed, yeah, I thought so. That's really interesting. Because don't you think that contact inhibition, as we know, plays a role in metabolic flux, for example, and in glucose uptake? Does it also play a role in ribosomal functions? Do you know that?
Well, I would expect that it will, because it is, I mean, ribosomes and translation responds to environmental cues, right? I mean, if we have a lot of nutrients, then the cell would respond by increasing the translation rate. So I would expect that.
Yes. Yeah, so I was curious about that, because if you could design an experiment, the dream question, if you could design an experiment, how would you set it up to mimic the in vivo environment optimally?
Yeah, well, so for the ribosome modifications or heterogeneity and specialization, I think it would be definitely nice to isolate ribosomes from freshly isolated chondrocytes, for example. They were not cultured. Mm-hmm. And also some experiments with expands, right? And like the plaques, for example. I mean, that would be quite representative of the cartilage in the body.
You would skip the culturing step?
Maybe, I will, yes. Well, that's definitely a next step. I mean, we're starting with the culturing because that is maybe the easiest to get all these readouts and do the experiments. But, I mean, if we want to understand how does it work in the body, I think that's next step to do.
Okay. Well, you're lucky in your field. You have these RBPM, what is it? The mutation, like the patient mutation.
RMRP.
Yes, that one. Which makes it very interesting to compare cells from these patients with like healthier controls. Do I have time for a final question? Yeah, great. I was wondering also about ribosomal biogenesis. On page 16, you write that it consumes the major part of cellular energy. And I was looking for the word metabolism in your thesis. I couldn't find it that often. And I was wondering, because TGF-beta also in our hands in fibroblasts, cardiac fibroblasts, plays a role in the metabolic switch from fatty acid oxidation to glucose metabolism. And I was wondering, does it impact that kind of metabolic decisions as well in chondrocytes? And is it related to ribosomal biogenesis? Do you know?
I would expect so. And also if we are in many of our experiments, when we are doing the RNA-seq, for example, we are seeing a lot of these metabolite-related proteins and molecules to be regulated or deregulated in osteoarthritic chondrocytes upon, for example, the TGF-beta treatment or osteoarthritic synovial fluid. So there is also, in osteoarthritis, there is this switch in metabolic network or the working of the cell.
That is really interesting. I think we can exchange a lot of experiences there. Thank you so much, and I give the word back to the prorector.
Thank you very much, Professor Schoen. The opposition will be continued by Professor van der Kraan, Professor of Experimental Rheumatology at Radboud University Medical Center in Nijmegen.
Thank you very much. Dear candidates, I want to congratulate you, of course, and your promotion team with its great work. I think it's a massive amount of work, very new, so you could be proud of it. And I was somewhat intrigued by your cover. At the first glance, I thought I was looking at a sketch from Escher. He's a famous Dutch printmaker. Maybe you don't know him, but it's good to go to a museum of him in The Hague.
Okay.
But later, of course, I realized this is the ribosome. But he's a very nice printmaker.
Okay.
But back to science, I would like to discuss to you mainly chapter 2. But I would start with some more conceptual questions. And one of the things which intrigued me is, what's the role of those ribosome modification? Is this a consequence of the osteoarthritic process, or is this also an initiating event in osteoarthritis? Could you speculate on this?
Well, thank you, highly esteemed opponent, also for the your compliment for the cover, and I hope that was a compliment.
It was.
Okay. So for your questions, you are asking about these ribosome modifications. Well, I think that they are drivers of osteoarthritis. So I think that, as I already said before, chondrocytes select for the specific ribosomes that are specifically modified during the process of development of osteoarthritis, and I think that these ribosomes then fuel all the processes, so the, for example, translation and expression of collagen type 1 related to fibrochondrocytes or fibrocartilage phenotype. So my opinion is that they are drivers, and yeah, they just drive and promote progression of osteoarthritis.
I haven't written it down, but a question pops up in my mind now. How about joint trauma and inflammation? Could it also be altering the ribosome composition?
Yeah, I mean, I would definitely expect that, especially because after joint trauma we have a lot of signaling molecules that are deregulated inflammation, right? And there's a lot of this signaling going on. So I would expect that maybe that is the point when the chondrocytes and ribosomes start to change. Because we have osteoarthritis is related to joint trauma, right? Very often that is a consequence. So maybe that is the starting point. And slowly we are selecting, or chondrocytes are selecting for becoming more and more osteoarthritic and producing more of these osteoarthritic proteins.
Okay, thank you. And I have another puzzling question, because I read on page 41 of your thesis that aging is correlated with the decrease in ribosome biogenesis.
Mm-hmm.
And if environmental factors reduce ribosome biogenesis, it extends lifespan. I found this hard to combine. Could you elucidate on this?
Yeah, it is a very, very variable. There are studies that showing that for lifespan, decrease in ribosome biogenesis is it is not preferential, but it is good for our, let's say, healthy aging. But there are also studies that are showing the opposite. So I think that there is a very complex regulation which we don't really understand at the moment. It's just very complex, and we really need to dive deeper, I would say.
Okay, thank you. Yeah, it intrigued me. It is said before, and you also showed in your presentation, that the composition of the ribosome or the biogenesis is regulated by TGF-beta. In some of your experiments, you use different concentrations of TGF-beta. And maybe you know that different concentrations of TGF-beta can drive different pathways, intercellular signaling pathways of TGF-beta.
Yeah.
Low concentration, mainly SMAD3, high concentration, mainly SMAD5. So you did not do this with the biogenesis experiment, or did you? Because I was wondering, is the biogenesis also different if you activate different routes? Do you know anything about this?
Yeah, that's a very good point. I mean, for this set of experiment, the first finding and the key finding was the boost in protein translation, right? The protein synthesis, in which we did test a different concentration of TGF-beta. But then for the ribosome biogenesis, we just selected the concentration at which we observed the highest increase in the translation rate, which was the 10 nanograms per milliliter, and then we just continued this experiment with this concentration. However, it would be definitely interesting to learn whether in lower concentrations we would see different effects, because as you said, like the concentrations of TGF-beta that is signaling through different pathways, and it could have very different outcomes. So it would be definitely something to look into.
Yeah, I also asked this because most likely low concentration are the physiological concentrations, and the high, the more pathogenic concentrations, so it has different effects. Another question that in most of your experiments you used either isolated chondrocytes, and passage 2, and a cell line. And you showed the effects. I was wondering, one of the things you noticed that the effect of TGF-beta took about 3 days. I was wondering, what are you altering? Are you altering immediately the metabolic setup of the cell, or are you altering the differentiation states of the cells? Can you speculate on that?
Yeah, I mean, very good question. And I think that also what we focus on in this chapter were ribosomes. And ribosomes, they can be regulated in a fast manner, like through different translation factors, which can dissociate with ribosomes. But then you can also affect ribosomes more slowly, so these ribosome modifications or core ribosomal proteins. So I think, and the 3-day stimulus is actually something when we can, the timeline in which we can measure differences in these kind of parameters, because ribosomes, the turnover time is about 2 to 3 days. So basically with this 3-day treatment with TGF-beta, we kind of turned over the ribosomal pool, and therefore we were able to measure these differences.
So it's related to the half-life of the ribosomes?
I believe that it is related to the ribosomes and their function and their modifications, protein composition.
Due to time, I have only one short question. You use TGF-beta 3. Why TGF-beta 3 and not what is commonly used, TGF-beta 1? These isoform, I mean, they are expressed by the chondrocytes and synovium, and they have receptors for all 3 of them. So they are maybe a little interchangeable, but also we tested TGF-beta 2, for example, as well on the rate of translation rate, and we observed the same effects on the translation, the speed of translation as with TGF-beta 3. So we tested these 2 isoforms, we observed the same effect. I mean, we could have taken IL-1 beta as well, TGF-beta 1 as well, but yeah. Okay, thank you very much for your answers. Very glad with it. I give the word back to the rector.
Thank you, Professor van der Kaan. The opposition will be continued by Dr. Staal. She's orthopedic surgeon at Maastricht University Medical Center.
Thank you very much. I would like to start by congratulating you and your team with this beautiful thesis. Thank you for your interesting work. I would like to talk to you about chapter 4. In this chapter, you studied the chondrogenetic differentiation of the growth plate and the McCusick type of achondrodysplasia. Yet in your impact paragraph on page 245, you conclude that, and hereby you refer to chapter 4, that snow RNA have a function in regulating the chondrocyte gene expression with consequences for signaling osteoarthritis. My question to you is if you can tell us what your opinion is of the relation between the chondrocytes from the growth plate of chapter 4 and those of the articular cartilage.
Thank you, highly esteemed opponent, for your compliment and also for the question. Okay, so the relationship between chondrogenic differentiation and osteoarthritis, I think there is definitely a relationship because the activation of the chondrocyte, which we observe in osteoarthritis, resembles the chondrogenic or the final phases of the chondrogenic differentiation, so the hypertrophy terminal differentiation. So I think there is definitely a relationship, and I think that what we found in the chapter 4 is that the snow RNA is really important. Snow RNA RMRP is really important for the for the chondrogenic differentiation and these terminal differentiation phases of chondrogenic differentiation. So I think that is how it relates to osteoarthritis. And also in osteoarthritis, there are several studies that are showing that RMRP plays a role in osteoarthritis, and it is increased in expression significantly in prehypertrophic and hypertrophic osteoarthritic chondrocytes. So there is, I think, definite parallel between these two processes. I hope I answered your question. Well, you look unsure.
You definitely answered the question, but what I'm still looking for is this. Is it the cellular change that has been made, whether the chondrocyte is in the cartilage or whether it's in the epiphysis, or is it more on a biological level, so the influences on the chondrocytes? What would your opinion be?
I think these are just changes in chondrocytes and their signaling, for example, and that they can lead to chondrocytes changing their behavior and going into this hypertrophic terminal differentiation processes.
So you think every cell of the epiphyseal plate could therefore be used for research in osteoarthritis?
That's what you mean. For different phases maybe of osteoarthritis, like healthy cartilage / osteoarthritic cartilage, I mean, there are definitely parallels.
Okay, well, thank you. We'll leave it for that. If we go further into this McCusick chondrodysplasia, there is a problem that is caused by the mitochondrial mRNA. The same mutation can lead to different phenotypes. So that means that there's people with short stature, which have the same mitochondrial RNA problem, but the phenotype doesn't have any hair or gastrointestinal problems.
Mm-hmm.
You have conducted research to the cells, and you describe them on page 106, and in table 3, you describe the influence of the research you conducted on probably the cause of what you think is the intestinal problem. Mm-hmm. My question to you would be, could you explain the difference in those phenotypes? So you have the same mitochondrial problem, and would you be able to benefit from patients having only the short stature and not the other clinical problems?
I will start with the first part of the question. So the RMRP has pleiotropic functions and can interact with different RNA substrates and cleave different RNA substrate. And these mutations are, of course, in different domains. And I think that maybe also yes, this influence, like what sort of phenotype are we observing in these patients? And then for the second part of question that was about the gastrointestinal problems, or like if you could benefit from different mutations of the RMRP in our research-ish.
Could your research benefit from having samples from the patients who only have the short stature and not the other phenotypes?
It would, or it could make the research questions and experiment readouts maybe a bit clearer and more like straightforward. I mean, that would not hurt for sure, but I mean, this is quite a rare condition. So getting some cells or material from this kind of patient is quite rare. So, I mean,
in the ideal world. Okay, can I ask one more short question? It's just a hypothetical question. In your chapter 2, I think it was, you only had male cartilage. Do you think there would be a difference if it would have been female cartilage? And can you elaborate on that, what the difference would be?
I mean, it could be. There are differences in like male versus female, and also the development of osteoarthritis. Also, I think hormones, right, could play some role. So, I mean, there could be an effect, but I mean, we are using articular chondrocytes, so maybe not too much.
Okay, thank you very much. I give the word back.
Thank you very much, Professor Staal. The next opponent is Professor Pruin, Professor of Biomolecular Chemistry at Radboud Universiteit in Nijmegen.
I think I can use this one. Do you hear me well like this? Okay, great. Thank you, Prorector. Dear candidate, first of all, I would like to congratulate you with the completion of your thesis. It's a very nice piece of work. And I also would like to congratulate your supervisor and co-supervisor. We do have some shared interests besides RNase MRP. I will not say more about that. We are also interested in biochemical processes in the synovial joint, although in my case it's more focused on rheumatoid arthritis and not on chondrocytes. But it's great to see that you sort of shifted research in this field to a basic biochemical process like translation, and I'm very impressed by the pioneering work you performed to address translational deregulation in a pathophysiological situation. Well, this is still early work in this field, focusing on these kind of processes, and as you already mentioned in your thesis, we are still missing many pieces of the puzzle, and I would like to address some of these pieces with you now. So particularly in chapters 5 and 6, you focus on the two types of modifications of ribosomal RNA, 2' methylation and pseudouridylation. And you exposed the cells to synovial fluid of osteoarthritis patients, and you identified a number of differentially modified sites, and these belong particularly to a group of so-called variable ribosomal RNA modifications. And you indicate that they may be involved in fine-tuning of ribosome function. Well, you also speculate that these modifications somehow may affect iris-mediated translation, and you provide some evidence for that. But as a biochemist, I'm actually asking what could be the molecular mechanism by which these very small, subtle chemical changes at the surface of a ribosome, how can they affect the initiation mediated by irises and messenger RNAs? Can you help me to understand this?
Thank you, highly esteemed opponent, for the compliments and well, for the question, and I'm gonna try. I hope I will give you some ideas. Okay, so maybe I will start that in one of these chapters I investigated the modification of a nucleotide 14 on 5'S. And it has been shown that modification status of this particular site can affect the conformation of the 5'S. And then 5'S is involved in translation and translocation, so elongation. So I think these modifications, they might be subtle, but I think they can have a small, as I said, fine-tuning ability. So like the conformation can change, the steric effect, or maybe some ribosome-associated factors now have a space to associate with ribosomes, and therefore, for example, initiate a translation from these iris elements. So, for example, we have this ITEF, so iris transacting factors, and in the TGF-beta paper, for example, we showed that they are less efficiently or less associated with the ribosomes. These ITEFs are less associated with TGF-beta treated ribosomes. So maybe that's also like a way how this iris-mediated translation, ribosome, PTM heterogeneity can be like correlated.
And do you think there may be specificity for irises related to the modification site on the ribosome?
I would expect that. I mean, the irises, each iris structures is a little bit different. There is not too much that they would have in common, or not like universal sequence. So I would suggest, or I would expect that that would be the case. Also, the ITEFs might be different for different irises. So I could expect that.
Related question I have is, I was actually asking myself, what will be the ratio between modified and non-modified ribosomal RNA, let's say in a normal cell, or maybe the cell of the cell line you use for most of your experiments. So will it be 10% modified, or 50%, or 99%? Do you have any idea about that?
Well, I think I know that the methylation level, basically that's what we are measuring with ribomethylation, or also pseudouracilation, that is C-score. They basically reflect on the fraction of the, so they reflect on, the fraction of the ribosomes that are modified at the given spot in the population. So all the ribosomal RNAs that we isolated, and then like ribosomal RNAs, if there are hundreds of them, or 100%, so all of the ribosomes in the pool are modified at this spot, then it gets the value 1. And then if it's the 20%, the value 0.2.
That's also true for the what you call variable modifications?
Yes.
Okay. Because then it would fit with the low level of iris-mediated translation in a normal situation.
Yeah.
Okay. But then it's a bit surprising that you do see in chapter 6 that when you look in the cells in which you depleted SNORA33, that in addition to the reduction of the pseudouracilation of the sites that's targeted by this SNO-RNA, there's also upregulation of pseudouracilation at other sites. But isn't that evidence that it cannot be 100% or almost 100% modification if you do see an increase? At least for these 3 sites.
Oh, you mean the effect on, for example, iris, that it's not 100% caused by the modification? Or what do you mean?
Well, there's an increase in the modification level observed, which is a bit surprising because these sites are not targeted by this SNO-RNA. That's not a question, but you do see an upregulation, which means that not all sites can be pseudouracilation in this normal situation.
Yes, that is correct. And I think that there could be relationships maybe between different positions that are not maybe that close together, but I mean within the 3D structure of the ribosomes, they definitely can like influence one each other. So that could also be the case.
So what could be the explanation for the upregulation at these sites?
Yeah, that's what I think maybe just said. I mean, the regulation of the modification at a specific site, it's not always so straightforward that we have a SNO-RNA change or changes in levels of SNO-RNAs, and then we see the same effect on the PTM. So for example, there are cell lines that have fibrillin knockdown or discorin knockdown, and certain sites were more affected than others, and these were mostly like the variable sites were more affected than the constitutive ones, and it was not related to snow RNA levels. So I think there are still some other regulatory mechanisms that definitely regulate the modifications of certain sites. And yeah, I believe that there are some relationships maybe between different modifications, and maybe it could be compensation, the ribosomes trying to compensate, but this is just, my suggestions.
Still a lot of pieces of the puzzle that can be addressed.
Yeah,
thank you for your answer, and I give the floor back to the program.
Thank you, Professor Pruin. Then the opposition will be continued by Professor De Bie, Professor of Research in Physiotherapy at Maastricht University.
Thank you. Dear candidate, I join the compliments of the previous assessors and opponents. Truly a remarkable piece of work, very detailed chapters, but very nicely written, and even for a layperson as me, a relative layperson, easy to understand. So big compliment on that, and it also shows in your presentation. Your presentation was really, really very nice. So I would invite you to give a lecture to our students, which is really, really good. I'm also relieved that despite what you write in your dankwoord, where you write you are a person of very few words, you managed quite a few, and successfully as well until now, so you're on a good track. When I read the articles and the individual chapters, I was also impressed by the multi-perspective view that you have and the different experiments that you do. I especially liked the article in which you use rare fibroblasts from the CAH patients, which introduces dwarfism, by your promoter also lovingly referred to as the mini-me's. But I had a question, but I understood you hate the Austin Powers movie, so I will skip that question. Now, but I'm still stuck with another movie, which is the Titanic, and you probably know the movie of the Titanic. It's a romantic movie, but it ends bad. And osteoarthritis also seems to end bad. And in the same line of research, you also have a colleague, Bas Hausmans, and he's there, but we won't mention him. But he discovered actually there are two types of patients, ones who have quick developing osteoarthritis and ones who have slow developing osteoarthritis. Now, as clinicians, you always wonder what to do with patients, and especially you start to worry when it happens quickly. Now, could you, based on your research, give me a clue what happens in those with fast and those with slow developing osteoarthritis? Because you say somewhere, and I was already very enthusiastic, you speculate about new possibilities for the development of treatments of osteoarthritis. So, wow, there you go. Help me. What should we do?
Well, highly esteemed opponent, thank you for the compliments and, well, some references and all your question. Okay, so the treatment of osteoarthritis and whether I can explain what is going on this fast developing and slow developing. Well, I think that also Bas found that, of course, the signaling pathways are all involved, and the signaling pathways also affect ribosomes. So maybe there could be some link in between those two groups, let's say. But I think that what might help is, of course, like the stratifications of these patients, and maybe in this book, I suggested that we could use snow RNAs maybe for this, to use as a biomarkers, either to detect the early stages of osteoarthritis, so then basically we have a chance to apply some sort of treatment or try to intervene. But maybe we could also use them to check the progress, how fast the osteoarthritis is progressing.
And sorry for interrupting you, but where in the process would you want to intervene? Because what I understood is kind of a cascade, and once one ribosome starts acting funnily, then the rest does it as well. So there's a signaling mechanism, and in some persons it's probably quicker than in others.
Yeah, I think that if we catch the early osteoarthritis, then what we also observe, and others observed it as well, is that in the early stages there is increase in protein synthesis, and this is not towards any like regenerative, or like it is not helping the chondrocyte health. and it's actually like detrimental. And if we could decrease the protein, the rate of protein translation to put it back into normal, let's say, we could, I believe, slow down the progression of osteoarthritis. And there are some, for example, mTOR inhibitors that have been shown in mouse and rodent experimental models to slow down expression or to slow down development of osteoarthritis. So maybe that would be something for the early stages.
Okay. And do you also think that, because we're still talking about slowing down a detrimental process, right? Do you also think it could be possible to reverse the process? Because we know that there are quite a number of cells in the body that are capable of healing themselves, right? Or kick out the bad cells, in case of cancer, and start anew. Would that be possible here as well?
Well, I think if we would influence chondrocytes to act healthy, even despite, I don't know, joint injury or early osteoarthritis. So, for example, I don't know, condition them with some SNO RNAs or knock down or overexpress certain SNO RNAs, we would make chondrocytes that are healthy, so they are translating healthy proteins rather than osteoarthritic. I think that in theory that could work to regenerative or regeneration of the cartilage, and rather than like repair towards more like fiber cartilage phenotype, let's say, to repair to more like fully functional cartilage, healthy one. I think that could be definitely something that would be interesting to look into.
Okay, now you're bringing me to a bridge, if I may still have one short question. Could I ask one of your paranyms to read proposition 11?
Let's do one last experiment.
Yes, and could you also mention who is the famous author of this?
Famous author Tim G.M. Welting.
Yes, so now, when you write something down like that from your promoter, then it must mean something, right? So what will be your next experiment?
Well, that's a very good question. Well, I'm switching for my postdoc a little bit, so I'm still gonna investigate ribosomes, but maybe in different disease. But maybe you were asking a bit more theoretical what I would be doing in terms of this thesis.
Well, you might answer both ways, right? But
Yeah.
we're losing you in the osteoarthritic field. Is that what I'm understanding?
For now, but I'm definitely going to be in close collaboration with the former lab, so I should not lose the touch completely.
Okay, I'm happy with your answers, and I give back the word to the pro-rector. Thank you.
Thank you, Professor Wie. Then it's time for the last opponent. It's Dr. Van den Beuken. He's assistant professor at the Department of Toxicogenomics at Maastricht University.
Thank you, Pro-Rector. Dear candidate, also I would like to congratulate you and your promotion team as well with this great achievement. And I was struck by proposition number 9 by David Bowie, which sounds like, I don't know where I'm going from here, but I promise it won't be boring. And I thought this statement is very applicable to your work, it's far from being boring. It was actually really a great pleasure to read it, and I think there are many more findings in the future in the field of SNO RNA biology and osteoarthritis, I believe. So having said that, I also have some questions before we run out of time. And actually, I would like to go a little bit more into some details of your experiments in chapter 5. So you use a bicistronic reporter system that is frequently used and also sometimes heavily criticized in literature, I think, in the past. And you can use this to actually measure cap-dependent translation versus iris-dependent translation, or cap-independent translation. And so in that figure, you report ratios of these two reporter genes, and then they're also even normalized to each control for each individual construct. And so my first question about this is whether you think by reporting these ratios and normalizing to individual controls, whether that might mask certain effects that you will not see in this normalized data.
Thank you, highly esteemed opponent, and for your question regarding the iris. Are you referring to the figure on page 143 or in, yeah, okay. So we definitely could be losing some information just by reporting the ratios, that's for sure. I believe that in a supplement I am mentioning the raw values. So in that case, like you can, or the reader can, of course, like check whether, or like what the values for each cistrand were, and where we can observe the effect on CAP-mediated translation as well, for example. But of course, we look at the data from individual cistrands, and we didn't see any effect on the CAP-mediated translation, for example. So then we just reported in the figure the ratios, which is reflective of the IRES-mediated translation.
Yeah, because I thought it was really neat to see this raw data, so I have to applaud you for that. I think that's the sort of transparency we need in science, that people actually report raw data. But actually, I also looked at that, and what I actually saw, which struck me a little bit, is that if you look at the CAP-dependent reporter, it seems to go up twofold for each construct after the addition of this synovial fluid. So what would that change your conclusions?
Well, we see increased proliferations in these cells. So, I mean, I would definitely expect that, yeah, maybe also the translation as all is increased. So,
right.
I mean, and if it was the same in all, then you can just look into the IRES-mediated translation and the ratios.
Right. So maybe the conclusion would actually be that overall translation is promoted, but
also increased.
IRES is even further promoted. Yeah. So that brings me to the next question about these irises. So the largest effect that you observe is for the viral irises. So they're not very relevant for humans in most cases. So the effect on a eukaryotic mRNA like p53. There, the increase in CAP-independent translation is actually quite modest. It's maybe 1.2-fold more after the synovial fluid. So that one makes me wonder, like, how relevant do you think that fold change is for the cellular biology? Can you speculate on that? Or?
Yeah, absolutely. I think in this study, what we were trying to emphasize was that the functionality of ribosomes can change. So maybe not which specific IRES elements are regulated, but that ribosomes can be fine-tuned, or like that these modifications, for example, can influence how ribosomes interact with mRNA, which I think these IRES data are showing that the interaction and maybe the reading through secondary structures of the mRNAs are somewhat changed. So yeah, maybe P53 and of course these viral IRESs are not too related to osteoarthritis, and it would be definitely interesting to look into the mRNAs related to osteoarthritis that have IRES elements. But yeah, just constructing these IRES construct bicistronic reporters is not so easy, but we are definitely working on that to looking into FGF1, FGF2 IRES elements. But I see that you want to ask another question.
Yeah, I'm looking at the clock, so.
Yeah, I saw that.
So also in chapter 5, you actually then look in a knockout pool of cells where you knock out SNORD71, and then you look at the effect on translation. And so one of the genes that you identify is COL1A1, and then you assess how it's regulated, right? So you observe nicely that the protein expression goes up by Western blot and LCMS, and you show that the mRNA abundance stays equal, right? So that suggests that it's post-transcriptional regulation. Right? And then you do this polysome assay. It's also one of my favorite assays. And so myself, I always struggle a little bit by interpreting how much the effect is on translation. And I completely agree with you that if you look at the amount of mRNA in the monosome, it's higher in one scenario. But yeah, I mean, in the highest fraction where the ribosome load is most, actually it goes a little bit against your prediction that COL1A1 is higher translated. So can you comment on that, or?
I think we look into that as a whole. So like if we look at the polysomal profile, in most of the polysomal fractions, the mRNA for collagen type 1 is more abundant in the SNORD71 knockout cell pools than in the controls. So Yeah, but it's always like the question of interpretation, and definitely we would be interested to look into the 5' UTR structure of collagen type 1 A1 mRNA to understand what is going on and whether this is really
You're almost saved by the bell, but I accept your answer. Thank you very much.
That was perfect timing. Thank you very much. Alžbeta Chabroňová, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Can't fly by now, don't waste all your time, 'cause I'll go the extra mile.
Alžbeta Chabroňová, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Welting is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
This is working. So, Alžbeta, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Alžbeta Chabroňová, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of our university. Esteemed Dr. Chabroňová, dear Alžbeta, congratulations with acquiring this great honor, the highest academic degree. These congratulations are for me, but of course also from Guus and Lodewijk, which made up your promotion team. My sincere congratulations also to your family with such a fantastic achievement, and super that you were able to actually attend Alžbeta's promotion in person. Now comes the exciting part, because I'm going to try to translate this in Slovak. Be careful. So, Vážená pani doktorka Chabroňová, milá Alžbeta, blahoželejme k získání této velké pokty na vašeho akademickou titulu. Tyto gratulaci se ode mne a samorečme aj od Gusa a Lodewika. I don't know what I'm saying. Moje upřímně blahoželání patří a vašej rodině k takému faštíkému úspěchu. Je super, že jste se mohli osobně zúčastnit na povýšení Alžbety. Teraz budem pokračovat angličtině. Is that okay? Okay. So now I'm relaxed. And stop sweating. So in 2017, we wrote a grant for a PhD student project for the Dutch Arthritis Foundation. And if you would get it, it would be the very first time that the osteoarthritis field would be looking actually at ribosome biogenesis. That is something you have been beautifully showing today. So we would be looking at ribosome biogenesis, protein translation, and the involvement of snow RNAs. That was a really long treasured wish that I had taken home with me to Maastricht from my own PhD research at the lab of one of your opponents, Professor Bruin, who is here today. So the idea of looking at the role of ribosomes in osteoarthritis was received with much enthusiasm and was actually considered very novel, and we got the grant. So the task that we had for the future PhD student would not be easy. Even we realized that. Ribosome research in osteoarthritis had never been really attempted. Almost everything was completely, almost everything was unknown, and a lot of the molecular biology techniques that were not yet running in our lab. But with perseverance, a super talented PhD student and our team, we, as in Guus, Lodewijk and me, were very confident to be able to perform the task ahead. So we went looking for a PhD student with a very strong motivation for molecular research and who would be not afraid of diving into the deep and exploring uncharted territories. So we put out a vacancy in the first months of 2018, if I'm not mistaken. And there were quite actually some candidates that responded, some really odd ones as well. Guus may remember that. But amongst those, there was this really interesting resume from you. And as a personal description of yourself, and I actually checked your CV that you sent me almost 5 years ago, you wrote about yourself, an ambitious, self-motivated molecular biologist with scientific curiosity and experience in the field, determined to achieve professional goals, flexible, cooperative, team player, independent, organized, focused individual, willing to learn, offering a passionate, positive, and proactive attitude, seeking a challenging PhD position. Well, I need not say more, and you can understand we found our match, right? So we invited you to start in the lab in May 2018. I'm not quite sure anymore whether you came by airplane or with your favorite Flixbus. But your travel brought you to Maastricht in the middle of the night. Since you could not enter your apartment, you decided to actually sleep on the pavement to be on time for our appointment at the lab at 8:30 the next day. And Alžbeta, if this was to further convince us that you were really dedicated to do the PhD, that was not necessary.
And I stayed longer.
And you stayed even longer, yeah. So, but issues with your housing and apartment were actually quite a red line through your stay in Maastricht. I remember electrical shortcuts, a major water leakage, and even bugs from pigeons on your balcony that somehow were infesting your apartment. But apparently, all these house owners wanted you to stay in the lab and work on your PhD research as much as possible. So, your PhD period started, and the first year we were entering some hiccups in the search for 47S pre-rRNA intermediates. So therefore, we expanded your research with snow RNA work with Professor Mandy Peffers and the RMP snow RNA in CHH cells. And these became really good chapters in your thesis. However, Guus and I were not satisfied. So the functional ribosome work would not yet really thrive, and we were trying to find a solution. You'll probably remember that. So this turned positively after a visit of me and Guus to Nancy in France, where I gave a lecture, and we met Yuri Moterin at the University of Lorraine. He turned out to be an expert in rRNA PTM detection, and one of his famous quotes, "You will get good data," turned out to be very true. And before we knew it, you were busy with 3 additional chapters that included functional ribosome work in chondrocytes and the mapping of rRNA PTMs. So with this work, you have truly unlocked a box of very exciting goodies and really generated seminal work that demonstrates how central the ribosome really is for the health status of the chondrocyte and osteoarthritis. And I'm convinced that this is only the tip of the iceberg, and we plan actually to expand that work in the osteoarthritis field even further. And literally, this is translational research. And I've always wanted to say that sentence because that is in our department something that we find really important, so that somehow our molecular research reaches the patient, but this is translational research, literally. So in order to do all this, make it to this day, and stand here proudly defending your work of the past years, you indeed kept your promise. That was written in your resume. I won't tell all these terms again, but I think it is due to all these qualities, I'm almost finished, that even despite the COVID disaster, helps you to get your PhD. And sometimes I wonder then, why all these experiments had these really funny nicknames, such as Please Kill Me Now, No More IREFs experiments, and I Need a Rope. So these were the names of her experiments. So I'm going to conclude. Alžbeta, I feel actually a bit sad that you're leaving, but I'm ever grateful that you choose our lab as a place to train yourself as a PhD and become an independent scientist in the growing family of PhDs that graduated from our beautiful laboratory for experimental orthopedics. It is fantastic that you found your that you found the postdoc position in the lab of our long-lasting collaborator and dear friend, Professor Peffers in Liverpool. And there, again, you will work on ribosomes, but now in the cardiomyocyte field, so that might even interest Professor Schroen. This is a great opportunity, and I wish you all the best of that. So I wish you a prosperous, exciting, and healthy future as a young doctor, and I'm convinced that you will make some amazing new discoveries in science. Never stop dreaming. Na zdravie.
Dear Dr. Chabroňová, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. I include your family, colleagues, and friends present here and online. I can tell you that the promotion committee was very satisfied with the way you presented your work and defended your thesis. You were very confident. At least you looked very confident, let's say it that way. And I hope you will look back on this PhD ceremony with a lot of satisfaction. I think you can, but of course...
do feel as well.
I would like to congratulate your promotion team, Professor Welting and Professor Van Rijn and Dr. Van der Akker. And I'd like to thank the promotion committee here present for being here and the time they dedicated to preparing this for this thesis ceremony is very much appreciated. Especially I'd like to thank our two guests from Nijmegen who came all the way up here, very much appreciated. It's always nicer to have the opponents live here in the hall than online, which happens often lately, but thank you very much for the investment of the time. With that, I will close this ceremony. It's time to celebrate now, and I wish you a lot of success in your further academic career. Close this ceremony.
Oh, sorry?